Hey everybody, Mr. Murphy here. I'm going to be talking about 2.1 to 2.2 in our textbook. I'm going to go through some of this a little bit quick, especially because it is pretty basic chemistry stuff. It's important to have a good background on it, but I'm not going to dwell on like every nitty-gritty detail about atomic numbers and stuff like that. Throughout the chapter, there's a bunch of different topics. So we start with chemistry, basic sort of understanding of why biology depends upon chemistry. Then carbohydrates and lipids, and a little bit about energy before we get into the next chapter where we dive more deeply into energy as it relates to biochemistry and enzymes. So this chapter opens with the question, why is the search for water important for the search of life? And you saw on the opening slide, there's like a picture of Mars. So water, being a very important polar molecule, is really essential to all of biology, and as far as we know, is an essential piece for living things. So just in terms of water, we want to make sure that we understand sort of water's made out of 2 hydrogens and an oxygen. It's 10 total protons and 10 total electrons, but because of the uneven distribution of electrons, it is polar, which just means that it acts like it has this positive and negative side. Well, it doesn't just act like it does. In fact, it acts like it's a charged molecule when it's not. So just a little bit of basics with chemistry. Obviously, just in terms of some words that we should remember, the nucleus is the center. That's the protons and neutrons, electrons around the outside. All living things are made out of these different atoms combined into a whole host of molecules, which allow for different functions and different things to happen. So some basic things just to remember, like charges will repel, different charges attract. This becomes increasingly important as we talk about protein structure in the next chapter. Typically, we consider atoms neutral unless they're in sort of a special case, ions and things of that nature. The elements are the fundamental, basis of what matter is made out of, and there's primarily 6 that we're going to be dealing with in this class, and that is carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. All right, so like I said, I'm going to kind of skip through some of the really basic chem stuff, but if that is something that you're having trouble with, you're certainly welcome to email me or dig a little more deeply into that. There's all sorts of YouTube videos that could give you like a really quick primer on what's going on with basic chemistry, or refresher of that if it has been a while for you. So with regards to sort of how chemistry ties into biochemistry and biology, we want to understand that atoms are things which have fundamental sort of desires. We use that word. Obviously, atoms don't probably want and dream of things, but it's helpful for us to talk about it that way in terms of filling electron orbitals and the tendency of atoms to make bonds. So the behavior of electrons is really what determines whether a chemical bond will form. And so here you see hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur, the 6 primary ones we talk about, and you can see sort of what their status is with regards to how many electrons they have in their different orbitals. For this class, for biology in general, it's important for us to remember that the first electron shell is going to want 2 electrons, the second is going to want an additional 8, and the third is going to want an additional 8 after that. Beyond that, as you probably know from taking a chemistry class, it gets a little more complicated, and sort of some rules get layered on top of that. It's, at the end of the day, the same basic gist of different atoms are sort of chemically and physically tending towards different electron orbital situations, and that is what governs really any type of bonds that are happening in any molecule. It gets a little more complicated, of course, because then you have different situations of temperature and pressure and all these outside external factors that can influence what an atom is sort of willing to do. But for us, following that 2, 8, and 8 rule is gonna be sufficient for us to understand what's going on inside of the body. So when atoms don't have filled outer shells, they tend to form bonds with each other, and that's what a molecule is. So just simple sort of terminology definition. And the octet rule, I'm never going to ask you about that term, but essentially what's happening when two atoms bond together, they're seeking to both fill their outside orbital. So a covalent bond, a sharing of electrons, is going to allow both atoms involved to either fully satisfy the sort of desire of filling that outside orbital, or be one step closer, so that, with additional bonds they can reach that point. So several different types of chemical bonds. This would be a really important slide to just have a good foundational understanding of. And on the right-hand side where it says bond energy, I will never ask you for these numbers. I won't even really ask you for like a comparison between these numbers beyond understanding which ones are very strong and which ones are particularly weak. In the case of covalent bonds, we have two atoms which are sharing electrons with each other. So each atom is sharing one with the other. So it is a much tighter and closer link together. Much more difficult to break, because in that case, both atoms are relying upon each other to satisfy those full orbits of electrons. An ionic attraction is where one atom has donated an electron to another. So sodium chloride salt is a really great example of this, right? Because the sodium atom has one extra electron beyond a full orbital. And chlorine, meanwhile, is one atom short of a full orbital. And so the relationship between the two is actually beneficial to both, that even though they become positive and negative ions, they are much more stable when that electron transfers from the sodium to the chlorine. Now, the problem is they're not actually relying on each other after that has happened. So once the sodium has given its electron away, it's not intrinsically linked to that chlorine. However, because the sodium is now slightly positive because it lost a negative electron, the chlorine is now slightly negative because it gained an electron. And so there is a just positive, negative, opposite charges attract kind of a attraction between them. And so you see it's a much lower energy than is it typically involved in a covalent bond, but still it's meaningful. A hydrogen bond is essentially the same idea, except that it's not actually between ions that have truly lost or gained an electron, but in this case it's against two polar regions of two different molecules. So remember, we said that the polarity of water is just the unbalanced distribution of electrons, and so there's a positive and a negative region. Well, that's the same thing here. A hydrogen bond, just to be clear, does not have to involve water molecules. Water molecules certainly give us a very prevalent example of hydrogen bonds. Further, a hydrogen bond does not actually mean the bond between a molecule and hydrogen. So this nitrogen here has two hydrogens that it is covalently bonded to. The hydrogen bond in this example is between this hydrogen and this oxygen on the right-hand side. So the hydrogen bond is simply the name for the polar attraction between these two things. And just to be clear, it's much weaker than a covalent bond. It's on the same level as an ionic bond. Beyond that, we have different types of just general interactions. And these are sort of the weakest forms of an attraction or a bond, but they're not negligible and they can't be completely disregarded. So hydrophobic interactions, that's just general interactions between nonpolar substances. It's not an attraction directly related to an exchange of electrons or anything like that, but just a natural tendency because of the distribution of electrons within these molecules for a very slight attraction. It's actually related and dependent upon this last one here, van der Waals, which is, again, just the fact that electrons at any given moment are flying all around a molecule. And so moment by moment snapshot of a molecule, you're not going to have an even perfect distribution. Even if that's a nonpolar molecule where you on average have your electrons distributed evenly and not in a polar way, just at a moment by moment glance, those electrons just by chance are going to be perhaps unevenly stacked. And that will lead to just a very, very, very slight attraction between molecules. So, quick summary, covalent bonds, super strong. Beyond that, ionic bonds and hydrogen bonds are weak, but meaningful if they're occurring in a high enough number, which hydrogen bonds we will see all throughout biology happening in very, very large number in the case of like protein structure, in the case of DNA structure. So it's absolutely important, but meaningful to know that it is much weaker than covalent bonds. So ionic bonds are just ions. We have cations that are the positively charged ones, anions that are negatively charged. Just some vocab that be vaguely aware of. It's not something that we need to be like making a Quizlet set and just make sure you know all the chemistry terms. There's usually enough information to support, but cations and anions, those are terms that come up often enough. We refer to any resulting bond between two atoms that are ionic bonds, that we call those salts. So it gets a little confusing. Salt itself, as like a food term, is the specific salt of sodium chloride, but there's countless other salts that can occur just because of an ionic bond between molecules. So here's that sodium chlorine example I showed you before. Generally weak, which is why they dissolve so freely in water. You know, the attraction between sodium and chlorine, for instance, makes sense. One's positive, one's negative. But when you put it in water and you're surrounding it by all of these other positive and negatively charged ends of water molecules, sodium and chlorine are like, see ya. Because they have plenty of other options of things that they could be attracted to that have the respective opposite charge. That's why they dissolve so well. Covalent bonds, super strong, like we said. So this is the actual sharing of these electrons. Here you see sort of like a representation of an electron cloud. Bohr models are great for understanding what's going on. They're not so great for understanding like what electrons actually do. Electrons are traveling at a pretty significant fraction of the speed of light. So thinking about them as these stationary little dots is helpful on like a diagrammatic standpoint, but not really close to a realistic representation of what they're doing. Carbon has 4 electrons in its outer shell, and so it wants 4 more, and that's why it becomes sort of the backbone for life. So here you can see a methane molecule, which is carbon and 4 hydrogens, and that really speaks to the versatility of carbon. It can make 4 different bonds, and so it can make these big long chains and these big interesting molecules that can therefore support much more complex sort of biological things. Some different structural formulas we're not going to need to, or structural models of chemistry, not something that we're going to have to dwell on. I think it's important just to understand that there are some more properties to these things that do matter for us. We're not going to really dive deeply into them, but certainly if you go on in college or beyond to take some courses in organic chemistry, you're going to get all sorts of details about how this precise orientation and bond angles and things like that really drive the specificity of these molecules, which then will further help you to understand on an actual application like biology standpoint just how specific the shape of things needs to be. And that's why we harp on that in the next chapter with proteins. So just to sort of wrap up some of this, the more bonds there are, the stronger the bond. That's why like nitrogen gas, which is 78% of what we breathe in every breath, doesn't do anything to us. It's triple bonded N2 gas. Our body doesn't have any sort of means or capability of tearing those apart, and therefore the nitrogen doesn't really react with us. We just breathe it in and we breathe it back out again. It's really just there to facilitate the oxygen being at the right percentage for us. Actually, that's kind of a backwards way of thinking about it. We've evolved to rely upon the nitrogen. It's not like the nitrogen's only there to help us. It simply was there, and we have adapted to doing okay with the correct sort of gas percentages that are in the air around us. The electronegativity, we'll talk more about energy and how it relates, but electronegativity is that attractive force that atoms are exerting on their electrons, and that's why when we look at water, we see that oxygen has a much higher electronegativity compared to hydrogen, which is why the electrons tend to stack on the oxygen side of the atom, and that then explains that polarity of water, that the electrons tend to spend more of their time orbiting on the oxygen end of the molecule, leaving the hydrogen end comparatively positive compared to the oxygen end, which is the basis of hydrogen bonds, which is the basis of water molecules, the basis of protein structures, or really any complex macromolecule structure. Anytime we talk about the different properties of water, cohesion, adhesion, surface tension, any of that, it all comes down to the very fundamental fact that water is polar, and that's it. So it is polar interactions and hydrogen bonds existing between water molecules. That is the only thing that drives all of those properties of water. More properties of water, more properties of water, properties of water. Okay, so certainly important stuff to just be aware of, but it's really just repeated examples of how that polarity allows for different and interesting things. The functional groups that we've gone over that are important to know: hydroxyl groups we see a lot, aldehyde groups we'll see periodically, it's good to recognize them. Same thing with keto groups. Carboxyl we will see frequently in our different monomers and polymers. Amino groups with proteins, phosphate groups with things like ATP and DNA is significant, and sulfhydryl we'll see in a very specific example of the interaction between different chains of amino acids that as they're folding up on each other for the 3D structure of proteins. All of this helps us to understand how the chemistry behind monomers and polymers comes together, and there are two key reactions that we'll discuss as we look at the formation of proteins, carbohydrates, nucleic acids, and lipids, and that is the condensation reaction, also known as dehydration synthesis, and hydrolysis. Dehydration synthesis or condensation is the combination of two monomers, so they are synthesizing a new molecule by dehydrating them. That is to say, we remove a water molecule, so an OH and an H actually comes out of this, and we combine these two monomers into a dimer here. We add another monomer and we get this little trimer. I don't think anybody actually says trimer, we just start calling it a polymer at that point. Hydrolysis is the precise opposite reaction. It is taking that water molecule and splitting it back into the OH and back into the H in order to break this 3 monomer molecule down into a 2 monomer one and a single monomer. Saying 2 monomer is kind of weird. A dimer and a monomer, and then further breakdown. So you can repeatedly conduct a hydrolysis reaction to break something down further and further and further, which is essentially the basis of what digestion is. All right, that's the end of this section, and next we'll look at carbohydrates.